My guest this week is Sigrid Jakob. Sigrid is an amateur mycologist located in Brooklyn and the author of a fungal DNA barcoding protocol that is circulating throughout the online mushroom science community. Like many of us, Sigrid is self-taught and holds no formal education in biology. Yet in spite of this, she's given lectures on mushroom identification, she's an avid contributor to the documentation of New York's diverse fungal ecosystem, and is really making her mark on the amateur community through helping others learn to do DNA barcoding from home. I believe it's people like Sigrid and the other curious self-learners out there who share a voracious curiosity about the world around them that can uncover that which is yet to be discovered. I'm intrigued by Sigrid's humility given everything that she knows, and it's truly a pleasure to give her voice more recognition.
Do it.
Well, Sigrid, thank you so much for joining. It's awesome to have you and to finally see you in the flesh. I want to share a little story about how I found you, if that's okay.
Sure.
So some months ago, before I knew anything about DNA barcoding, I was browsing the shroomery.org forums. And for people who are not familiar with the mushroom world, Shroomery is probably one of the oldest mushroom growing forums, very focused on psychedelic mushrooms though. But they have some other sections for like general mushroom identification. And a person made a post about, hey, how does my gel electrophoresis results, how do they look? And I didn't know anything about that. So I took a look at his post, and nested deep several pages within the thread, he mentioned the protocols that he was using to do DNA barcoding. And one of the links was to this random Google document that I was like, okay, first of all, how did he find this Google document? Because the other one was Alan Rockefeller's protocol with the counterculture labs. But I didn't know, I was like, what is this document? And then I read it and I was like, this is actually probably the easiest version of DNA barcoding that I've seen. And I think I could do this. I had also found Alan Rockefeller's videos and his write-up. He did a big thing on Facebook several months ago, maybe 6 months ago. And I remember watching those and going like, oh, that's so cool. I didn't know anything about it. I was like, wow, I could actually do this at home, which is pretty cool. Extract DNA from mushrooms and so on and so forth. So I'd already been kind of thinking about it. But then when I saw your protocol, I was like, you know what? This is actually easier than Alan's. No offense to Alan, because there's probably some trade-offs that I'm not even aware of. But this is actually even easier and very easy to follow, easy to read. And your protocol is actually what I use to do DNA barcoding for the first time. I have some PCR products sitting here getting ready to go to the lab, and I'm planning to use your protocol again. And you and I have exchanged notes on some of the things that I encountered while I was using it. And I'm going to do it again this evening. But so you're the author of that. And a couple of weeks ago, the gentleman, and I can't remember his name, I should really know because he's really popular on Instagram, but the guy that runs the Mushroom Hour podcast.
That's right, yeah.
He gave you a shout. He gave you a shout out as well. And I was like, wait a second, this world must be really small, because I think he was mentioning something about you telling him about your protocol as well and him recommending it. And so, yeah. So anyways, you've written this protocol for DNA barcoding that I think is super beginner user friendly. I think is going to introduce a lot of people who are into the growing world of mushrooms and mushroom growing to the idea of the genetic side, which is fascinating in my opinion. And so it's really a pleasure to meet you and to have the author of that protocol here. So thank you. So thank you for joining. Perhaps we could just start by, you could just introduce yourself and talk a little bit about who you are and your backgrounds. And
Right. Okay. I live in Brooklyn. I'm 54 years old, mother of a teenager. I'm not your typical sort of biohacker type at all. I was born in Germany. I'm British, American. I make a living as a strategist, as a freelance strategist. I help large companies solve their biggest problems. So why am I doing this mushroom PCR stuff? God knows. I have no idea. I stumbled into the world of mushrooms about 4 or 5 years ago. I joined a mushroom club. I go out every weekend, look at fungi. And I got really frustrated when I find something that's not in the field guides, that I know is probably special, but you ask people, you know, what is it? And nobody can give you an answer. So the only other thing you can really do is like you barcode that mushroom. So I did all the usual stuff. I looked at Alan Rockefeller videos on slow motion, taking notes over and over again. But it seems super intimidating. I mean, I studied philosophy, psychology, and photography. So I do not have a scientific background at all. And I always went like, this is probably way out of my league. And actually, when I started posting in some of the forums, people told me like, girl, you know, this, you're in over your head. This is not going to work. And I went like, damn it, you know, I'm going to give it my best shot. And so with the help of a couple of mentors, I actually managed to make it work. And then I went like, okay, I'm going to write a protocol for people like myself, i.e. people who know nothing, don't know any of the language, you know, are totally intimidated. I'm going to write a protocol so those people can also get access to that technology. So yeah.
Wow, that's an incredible story. And I love talking to people like you because we are DIYers in the truest sense. We're self-taught. We're just taking information that we see on the internet and we're trying to imitate it and learn about it. And but I think it's a great way to learn because look how far we've gotten, you know, just by doing that. And also there's just so many haters online. I hate the fact that there's people out there who don't even know you. They don't know what you're capable of. And they're telling you, no, you're way in over your head. And I bet most of the time, 9 times out of 10, they've never done it themselves. So they have no idea what they're talking about most of the time. But you persevered, and that's par for the course with interacting with people on the internet a lot of times. So how long, I want to get into the DNA barcoding because I think a lot of people may not even be familiar with what it is, and we'll talk about that. But I want to kind of just go back into your personal history. So you don't have necessarily a background in science, neither do I necessarily, although I come from the tech world, but you come from the media marketing, strategic corporate world, I guess you would say, perhaps, or consulting.
Yeah.
So that's, how did you first get started with mushrooms, and what made you interested in them, and what were some of your first experiences with getting into the world of, you know, fungi?
Okay, well, I grew up in a small village in Germany near the Black Forest. So there are mushrooms out there, and there's a history of picking them. So we did that when I was a kid. And I remember seeing a lot of stinkhorns in the forest, and I was always super fascinated by like the weird sort of almost psychedelic sort of looks of some of those fungi, and that really stuck with me. But then I didn't do anything with that interest at all. I moved to New York City 24 years ago, and I always went like, I'm in New York City, there's no mushrooms here, will be a hobby for my old age or something. But then I started seeing them, and once you see them, you can't unsee them, and suddenly you realize there's fungi everywhere. And that's when I joined the Mushroom Club about 5 years ago, because I went like, I want to know more about these strange fungi. And we found over 1,000 different species in New York City. So, I mean, there's just tons. It's one of the richest fungal ecosystem in the world, and they're just literally everywhere. So, yeah, and then I just got hooked because it's a community. You go out into the woods, and it's fun, and, you know, it's very rewarding when you find something rare. So it's a really nice counterbalance to the corporate life.
Absolutely. It keeps you grounded and connected to nature, especially in a place like New York City, where that can be in short supply at times, I would imagine.
Yes.
So what are the biggest differences? You know, I don't know how long you've been in the US compared to Germany in terms of like mushroom stuff, but did you have that connection as a child? Like, did your family and you used to walk through the woods and do mushroom hunting, or like, was that later in life that you got interested in it?
Well, I got forced to walk in the forest every weekend because my parents had a nursery and they were obsessed with trees. And my family has like generations and generations they were all foresters basically. So they're forest people. So my mom and my dad would drag us into the forest and like talk in Latin names. Everything was in Latin names. Wow. And we just went like, bloody hell, this is so boring. Have to do that. It was like pure torture, and you know, and I really didn't enjoy it. And you know, we sometimes find fungi and we eat them. That would be the only good thing. But I really just resented that whole thing, and that's why one of the reasons I ran away when I was a teenager was just like I really, really hated small town life with all this boring nature. So I was very much a rejecter of all that. And now I'm going like, that was such an idyllic childhood. And I was so lucky, but I didn't see it like that at the time at all. And now I'm that boring mom, you know, like I'm boring my teenager to tears with my Latin names and all that stuff. So it comes around.
I would imagine that experience gave you some kind of advantage when you rediscovered your passion. You were able to reform those connections with the memories and even the nomenclature that your parents used to use for describing species. And did you find that was gave you some advantage, at least in terms of just starting completely from scratch?
I want to say I have an easy time remembering Latin names, for example. Like I have a large capacity to absorb Latin names, and it must have been hereditary. So for sure. And I definitely still have some of that tree knowledge, because obviously fungi and trees are very much connected. So you're always trying to figure out what trees, what growing under. So I do have that memory bank, for which I'm very grateful.
I'm always amazed at the people that can just go out in the wild and see a mushroom and go, yes, that's Amphalatus nidiformis or whatever, you know, they could just like say it off the bat. And there seems to be no limit to the extent to which they can just rattle off hundreds, if maybe not thousands of species names that they have by heart. How do you get to that level? Like, how do you develop? Is it just practice and repetition and just constant researching and reading? Like, how do you get to the point where you're that familiar and comfortable that, one, you can identify most of the common fungi that you would see in the wild, but just remembering that many names?
Okay, I guess my secret is iNaturalist, the app, because like it's sort of like almost like my flashcards. Whenever I find something, I log it in iNaturalist, and so the name sort of begins to ingrain itself, and then you go over those finds once in a while, and you know, the name gets triggered again, and then next time it somehow just pops into your head when you see something. You say Nibio fragrare, like it's nothing, and yeah, so iNaturalist, that's my secret superpower.
And iNaturalist is a website for folks who encounter species of wildlife, flora mostly. I don't think there's animals on there.
Oh yeah, it's everything that's alive.
Okay.
Yeah. And I just want to like do a little commercial for iNaturalist, because citizen science, you know, a lot of people talk about citizen science, and this is the best tool to sort of make an observation of all the nature that's around us. Oftentimes it's threatened nature, or, you know, with mushrooms, they're very sort of evanescent. They're only there for like a day or two. So it's just a wonderful way of capturing all that stuff. And scientists work with it, mycologists, conservationists use that data. And it's just a wonderful way to, you know, contribute to science, even just very casually. It's very quick. It takes like, 10 seconds to make an observation. Plus, people will ID your stuff. So if you don't know what you found, somebody else will tell you what it is.
So how does it work if you're just a newcomer and you're not familiar with it and you don't necessarily want to go the scientific route? How does it work? How would someone who wants to start using iNaturalist begin to take best advantage of that website?
So it's super easy. It's an app. So you sign up for it. That takes, a minute to do. And then next time you see, a mushroom or a tree or a frog or whatever, you switch on the app, you take a picture with it and you upload it, and it'll instantly generate a suggested name. And oftentimes that's already correct. And then you'll go like, that's the name of the butterfly I just saw. And then you've made that observation. It takes 30 seconds and you already know what you found. Or sometimes with fungi, they often look weird. The app doesn't know what it is. So you just put in fungi, and then somebody else will come along, like an identifier like myself who does it for sport, and go like, actually what you found here is X, Y, and Z. And it's really remarkable because it's quite rare. So, it's very easy.
Yeah. Well, I also find that there's a sort of satisfaction that you get from, like, once the bug hits you, you sort of get hooked on it. Like you want to capture as many new organisms, especially things that people don't see very often, kind of rare things, and get them up and then try to figure out how to identify them. And there's a satisfaction I find with kind of building a portfolio of your observations of things that you found in your natural environment.
Well, it's Pokemon Go with the real world. And honestly, it's also 200,000 years of engineering, because like we've been selected for spotting things in nature and identifying them. We're the ones who are smart, who were able to survive using that knowledge. So it's a human instinct, it's a survival skill. And no wonder our brain finds it rewarding, because it's a very useful thing to be able to do.
Yeah, absolutely. That's one of the side effects that I kind of think about with masks sometimes, is that it kind of shields our faces. My wife had an experience where she went to this is not really related to fungi, but she went to a grocery store, and she was in the checkout line, and she looked down, as we normally would do, to a child that was waiting with her mother in the checkout, and the child couldn't see her face. And the mother misinterpreted my wife, like looking at the child, like as if maybe she was like being rude or something to her, but really she was trying to smile at her, but neither side could see each other and see the intent. And I just thought that was like, it was a little sad to see that kind of side effect of that. But I don't know why I shared that story. It just kind of reminded me of like what you were saying. We've evolved. We definitely have neurons in the brain that are specialized to recognize faces and emotion. But I believe those are probably very similar to the ones that are used to recognize species of birds, species of fungi, species of, and we can really fine tune those muscles with practice, I find as well. If you just pay attention, attention. And
and it gives you that little dopamine hit every time you do it. Like, it's so rewarding to be able to identify something. It's very addictive.
Yeah, absolutely. So, if you get into mushroom hunting, eventually you're going to encounter a species that you just aren't able to identify. And maybe you could talk a little bit about what are the initial steps that you take to try and identify a mushroom outside of DNA. What are the ways that you could kind of narrow it down to try to figure out what the organism is? And then when does that begin to fail?
Right. Okay. I mean, there's this mushroom that grows in my local cemetery that comes back every single year. And I know it's a Cordinarius because it has like all the Cordinarius characteristics. It has a cortina, which is like a spider web around the gills. And then you can get a Cordinarius guide, a book that essentially is a dichotomous key, where it asks you always two questions. Does it have brown gills or does it have white gills? And it's so you follow that chain along, and ultimately the idea is that it takes you to a species if you answer enough of those really specific questions, and you look at all the morphological sort of characteristics of all. What does this stem look like? What does the spore print look like? You look at it under the microscope and look what's the shape of the spores and the size of the spores, and then oftentimes they'll take you. You follow those breadcrumbs and eventually it'll take you to the right destination. But sometimes a species is really rare, or you've taken the wrong turn, which also happens all the time. Is that brown or is that gray? And you make a choice and it was the wrong choice. So that definitely would be your first way of attacking the challenge, because you don't always want to spend the $4 or whatever it takes to do the sequencing. That's lazy. You need to be disciplined about that stuff. And I try to be, but sometimes you just, like with that particular mushroom, I just never hit pay dirt. It was always like it petered out somewhere, and the keys never took me to anywhere. So, I finally had to do DNA on it, and it turns out it's a really rare Cordinarius that only grows under linden trees, and it's called Cordinarius balteotucomatalis. It has a very, very long name, and it's only been found like 5 times or something. And that's where sequencing is really helpful, because I just would have never in a million years have gotten to that species, because it's not in most field guides, and
So if you're a new mushroom hunter, there are reference manuals and field guides and plenty of online resources that will give you all of these morphological characteristics of mushrooms that you mentioned, and you use those as references. So you make an observation of the mushroom that you found, and you compare it to the references. And that's what will help you narrow it down, in addition to the apps now that use sort of an AI image recognition to kind of take a lot of that guesswork out for you. But so when do you also need to complement the physical morphological features with, say, looking at the spores under a microscope? Because my understanding is there are a lot of mushrooms that are very similar with their sort of macro morphological characteristics, but you then need to look at the spores as well to make a differentiation.
Absolutely. Yeah, that's a really important tool. And one of the reasons I have two microscopes is to do just that. And it's not just the shape of the spores or the size of the spores. You also drop them in reagents like melters, which turns it specific colors or doesn't turn it specific colors. So there's all these fancy other things you can do. And that's very useful with amanitas and certain other types of fungi. It's definitely one of the sort of best ways of getting to a good ID. I will also say that there's gonzo methods that I sometimes use where I literally just Google something. You find like a tuber that smells really bad and you can't key it out, and you just like Google, bad smelling tuber until you find something. And sometimes that works too. There's also all these mushroom forums, some of them like Alan Rockefeller, it will contribute, and people who really know the stuff will contribute, and you just post a bunch of images and maybe images of the spores as well, and then people will give you an ID. So that's another resource that I actually use quite a bit, to my embarrassment.
No, I do the same thing. I mean, there are some really knowledgeable people that you and I are aware of out there that can just see a picture and instantly kind of know right away what it is, because you've probably seen it a million times. So even after you look at all of those morphological characteristics, you've looked at the spores under a microscope, there's still some cases where there's either not enough information or ambiguous information, and that's where the DNA barcoding can come into play. So let's think about talking about that as if someone's never heard of DNA barcoding before. How do you describe it to them?
Okay, so yeah, it's going to have to be a simple description because, like I said, biology is not my thing at all. So the way I describe it is like every single organism has like a user's manual, how to build that organism. And it's as thick as the New York City phone book, what DNA barcoding is, instead of looking at the whole phone book, there's usually only a very sort of small area that actually where there's a lot of difference between organisms. So you're only looking at a very small section, and that's called the ITS region. So in the IT, because a lot of the stuff when you look at two organisms, it will be the same, the sort of basic sort of biological functions. But that little region, there's a lot of difference, and that difference has been systematically mapped. So if you take an organism and then you take just a tiny little piece of it and you smash the DNA out of it, because DNA does not like to get removed from an organism, because if it did, that would be bad. If it rained and all your DNA came out, that would be a disaster. So DNA really wants to sort of be in the cell. So you're going to have to use heat, you're going to have to use chemicals, you're going to have to use force, you're going to have to use all kinds of things to try and get that DNA out of the cell. And that's sort of the extraction portion of the PCR process. If you're successful, you have a tiny little bit of DNA in your tube, like just a few bits, and that's not enough to do the whole process. So what you need to do is you need to amplify the hell out of it. You need to take those two pieces and turn them into billions of pieces. And there's this amazing process called amplification, where you systematically heat and then cool and then keep it at normal temperature and do that 35 times. And every single time you do it, it doubles. So if you do that 35 times, you end up with billions and billions of pieces of DNA. And then the last step is really, you want to test that to make sure it actually worked before you spend your $3 or whatever. That's called gel electrophoresis. And then you send it off to a lab for Sanger sequencing, which is very quick and very cheap. And they'll send you a string of letters. And that string of letters is that small region in the overall sort of larger phone book that differentiates your organism. Then you put it into something called BLAST, which is Google for DNA sequences, which is very easy. Anybody can do it. And it tells you what sequences it most closely resembles. And if it's like 98% close to this or that organism, you go like, that's probably the same organism. So yeah, that's how I would describe the process to someone who knows nothing, i.e. myself.
No, you know a lot more than most people, I would say. And I like to hear the non-scientific, proper version, because we're trying to democratize this information and make it as accessible to as many people as possible. And I find you have to sort of get people hooked into the idea of what it is. And then through the process, you will do digging in, begin to read the papers and begin to learn the underlying science behind it. But maybe we could go through just each step and talk a little bit more in depth about it. So the first thing you said is there's pretty much every organism has these, every organism has DNA. I think we all kind of know that, but there are sections of that DNA that I believe is, in particular, the ITS gene that you mentioned is a non-coding region, meaning that it doesn't actually get transcribed by the cells in the whatever the organism is. It's part of the what's called the ribosomal DNA, which is the ribosome that's in the cell itself, is what actually grabs the DNA at 3 nucleotides at a time and turns those into amino acids, which get turned into proteins, which gets turned into more complex tissues. But there are this region that scientists have discovered in fungi. I don't know that it's in animals as well. I can't recall exactly. There is a different region in bacteria, but by and large, there's about, my understanding is about 3 or 4 genes used across all living organisms that can be used to compare the sort of the family tree of life between all of those organisms. The one that's used predominantly for fungi is the ITS gene. Again, it's a non-coding, and then the ITS stands for interspace something or other. I can't remember the exact name. Doesn't, you don't necessarily need to know it, other than it's just, hey, there's this gene hanging out in all fungus, and it's it's in a known location, which that's kind of important, because you kind of have to know where that gene is in relation to all the other genes so that you can extract it out and amplify it. So the first step when you do DNA barcode is you obviously need to either culture or collect a specimen so that you can take some DNA out of it, yeah?
Right, yes. And the small piece, people always imagine you need a giant chunk of fungus. It's literally like 1/10 of a pinky nail or something, yeah.
Yeah, I'm surprised. Like, I have these little Eppendorf tubes, and you really, I think one protocol says a grain of rice. That may be yours. It says a grain of rice, but really
much less than a grain of rice.
Yeah, you really almost like a pinprick, because you really, from a molecular standpoint, you really only need 1 strand of DNA.
Exactly.
So that's incredibly small. So you don't need a lot of it. Then I think in your protocol, we mix it with sodium hydroxide, I believe.
That's right, yes.
And that will actually break the cell open and allow the DNA to, in solution, basically, allow the DNA to kind of escape the cell. Then we centrifuge it, which basically allows all the particulate matter, all the cell matter to pelletize at the bottom of it. And what's left is this clear liquid, a very small amount of it, actually. And you go, I remember I did this the first time. I go, I don't know if this worked. I mean, how can this is a really small amount. Is it really in there? It's like,
yeah.
There's so much of it. It's like magic when you're doing it. You're just like, all right, I'm going to put this very tiny drop of reagent in this, and hopefully it's actually enough. So yeah, that's really the extraction part of it. But the next thing you mentioned was like, okay, you've extracted it. Now you have all of this DNA, but you have the whole DNA, right? You have the whole DNA from the fungal cells.
That's right, yeah. So you need the primers to cut it, to snip it off at just the right points. You put in a forward primer and a reverse primer, and that tells the reaction to only amplify that small bit of the DNA, not the whole phone book. So yeah, that's very important. I completely didn't talk about that.
No, I like and those primers, because this is a relatively new concept for people who aren't into DNA or genetics. Right. But these primers are these, they actually are just short snippets of DNA themselves, but they're aligned just before and just after that ITS gene region that we want to take out. And then there's another enzyme that gets mixed in that actually is responsible for like cutting it all out. And then, yeah, the PCR, which is polymerase chain reaction, we won't go too deep into that because people will probably fall asleep. But the idea is when you take this DNA, you're going to mix these primers in, you're going to mix this enzyme, which is the polymerase, Taq polymerase, which actually is pretty cool. It comes from a bacteria that came from Yellowstone and can survive really high temperatures. Mm-hmm. But then you're just going to heat the DNA up and lower it a bunch of times. I think you said 35 times.
Yeah, that's about it. Yeah, and that's it.
Yeah.
And suddenly you got billions and billions of strands of DNA, of that little snippet of the DNA. Yeah, it's crazy.
Just that one template, I think is what it's called. So one little piece that you extracted just heating the DNA up and cooling it down, this amazing chain reaction happens, and you end up with a bunch of copies of the one that you need. So why do you need a bunch of copies is a good question. And
well, I guess the lab wants a lot of needs a lot of it to do the Sanger sequencing on. I mean, I don't really know, but I guess more is always better.
I think that I don't know a lot about sequencing, but I think it actually has to do with something to do with the sequencing. I mean, I think you need the more that you get, and the better the quality of it is, the more accurate, precise your sequencing will be.
Right. That's right. Yes.
So you go through. So you do the extraction, you do the PCR. Then the final step is something called gel electrophoresis, which I must say is probably my favorite step.
It's so cool. Yes. It's so rewarding.
Maybe you could go a little bit deeper into like, what is that fancy word, gel electrophoresis?
Right. It's just a test. I mean, that's what it is. And you basically just pour some agar into a container, and agar is just, I guess, an algae, and it makes this sort of weird jelly thing. And then you pour a little bit of tris buffer over it, and you put the DNA into these little wells that you put in there with a comb. And then you put a current through it, and what happens next is like the DNA gets sort of dragged through the gel, if you put a little color into the DNA, and it shows up as bands. And if your whole process worked, you get these beautiful little bands in the gel. And if it didn't, it's just going to be a blank lane, and it failed, so you can save your money. But yeah, that's sort of the basic principle. But honestly, I'm the worst person to explain these things, because I really don't know how to do it, but is that actually what happens? I hope it is. I think that's what it is.
No, I think it's a great explanation. That's exactly right. I think some, like, people who've never done it before, we've all sort of seen these pictures of bands and people going, hey, that's DNA. I remember I used to see these black and white photos of DNA, the DNA bands, just in media or just like maybe in a textbook. But I never understood how they, like, what is that? Like, how do you read that? What is it? And it's really, like you just said, it's a picture of a band of DNA. And because DNA is negatively charged, so the gel electrophoresis, like you have this gel that's in this box, I guess you would, or tray is going to be in. And there are these electrodes. There's like a negative end and a positive end. And when you give it power, there's a current running through it. And because DNA is electrically charged, it will begin to move through the gel. Now, the longer the piece of DNA is that you extracted, the more negatively charged it will be. And so that determines how far that band moves up or down the gel. And so they have these other things like DNA ladders, where you can kind of compare where the band went in relation to the DNA ladder to kind of understand, like, exactly how big was the DNA that I extracted in PCR. Did I actually extract the ITS gene, which is of unknown size, or did I make a mistake somewhere? And did I, we're not designing our own primers, but there are people that design their own primers. Actually, you may design your own primer.
I've done a little bit of customization of primers, but yeah. But I mean, the whole point is, like, you're just saving a bunch of money, because you could just send the output from the PCR straight to the lab and save yourself the whole gel electrophoresis step. But the thing is, stuff doesn't always work. So you only really want to send off stuff that has proven to work. So that's really only why we do that stuff.
Yeah. So if you see the band and the band is, looks like a good band, then for the most part, that you've done everything correct up to that point, and you can send it off to the lab.
Right.
So you send it off to the lab. What happens after that?
Well, they do some magic on it, and then they will send you these files. You get these AB files and all kind of fancy files, and you open them up, and you get these beautiful chromatograms, different colors, different sort of wave shapes. And that already gives you kind of an idea of whether you had a good reaction or not, because if it's like weird and squiggly and they're all on top of each other, that's usually bad news. And if they're beautifully and even and just like that, then that's usually a really good sign. And then it's really just a visual way of displaying the letters, because ultimately what you're going to get out of it is just a sequence of letters, like A, C, T, G, ad nauseum, usually between like 300 and 800 of them, and that's your sequence. And that sequence you can just plug into the BLAST search engine, and it tells you what you got there. But yeah, it's, what you get back essentially is just a bunch of letters.
Do you have to do anything with it? Like I know on Alan's video, he cleaned it up a little bit.
Yeah,
I need to go back and look at that, but could you talk a little bit about what that is?
Sure. The beginning and the end of a sequence is usually messy. It's like carrots. You kind of have to top and tail them. You know, like you chop off the front where it all looks messy and there's lots of ends, and the back is usually really messy. But you could just, when you have the actual letters, you could just like start where all the ends finish and then stop where all the ends begin. N is bad. You don't want any ends. So you just want the clean stuff, and you can just copy paste that part of the sequence. It takes like 2 seconds.
And it's not going to make a big difference, because I think what's happening is when you run the BLAST tool, it's running an algorithm that compares that sequence against all the known published sequences, right?
Yeah, and honestly, if you leave all the crap in, it mostly makes no difference. I mean, it gives you less of a higher number in terms of percentages. Instead of 98% similarity to the sequence, it says 96 or whatever. But yeah, it's easy and it's not that important.
And the BLAST tool is on the National Institute of Health website. Does it, when you run the BLAST tool, does it tell you right away, hey, we recognize the species of this sequence, or you know, what does the result tell you when you run it?
Right. Yeah, it's not like Google where it just tells you all the answers. It just tells you what sequences it's similar to. You know, it says like, well, it has a 98% resemblance to this particular other sample that somebody entered into the database, or 95% resemblance to this sample. And it also tells you the amount of overlap between your sequence and that sequence. Sometimes it's only 80%, so because your sequence was maybe a little bit short, or there just wasn't that much overlap. And it assigns a quality score, and that quality score ultimately drives what's at the top of the pile. And then you can take a bunch of those other sequences that yours is similar to and create a phylogenetic tree. So you're essentially creating a family tree for your sample, where it tells you all the other siblings and cousins and remote aunts and uncles of your particular sequence from their database, which is also really cool because it gives you a sense of how this fits into the larger universe of other fungi. So, and it's really easy. Yeah, it's super easy. Like, yeah, I can do it.
Yeah, that's pretty amazing. So you can go out and find a piece of fungus that you tried your best to identify using all the physical features. Then you can extract the DNA, the ITS gene, you can send it off for sequencing, you can run the BLAST tool, and now you have a pretty high degree of accuracy on what the species of that is. And I would imagine that combined with all of the other data that was collected, it really kind of builds a really high degree of confidence that you have indeed successfully identified this previously unidentifiable organism.
Well, that's one scenario, but the other scenario happens just as often, which is like, oh, you got like 93%, which is already not, that's your closest resemblance. So you go like, did I just discover a new species, or was it just a crappy, a crappy sequence? And the thing is, there's tons and tons of undiscovered fungi out there. I mean, it's estimated there's 5 million species of fungi. We've described about 120,000. So the chances of finding new species are actually shockingly high. So it does happen quite frequently. But so in theory, you could go ahead and start research that new species and, describe it and name it after your girlfriend or whatever. So but sometimes it's also just like not a great quality sequence, and sometimes people just haven't put in a lot of reference sequences into the database. Like there's not enough raster type specimens in the GenBank database, so you have nothing to compare yours to, even though out in the world that species exists. It's not in GenBank. So the last thing I will say is like the thing you can do for science is you should enter your sequences into GenBank as well, so that future scientists can learn from your sequences. So you add your iNaturalist reference to it, so people can look at the pictures and the descriptions. And then the next time somebody finds something similar, they can go like, oh, somebody found something just like that. So you're giving back. That's really important.
Yeah, and I was just thinking maybe that's something that we could work on adding to your protocol, is that the post sequencing procedures, how to do the cleanup, how to run blasts, and how to publish the information to
Right.
GenBank.
And Alan has some lovely videos. The things I do not want to compete with Alan Rockefeller. He's amazing. And I would hate to just sort of do what he does. And yeah, because he's a total hero of mine. And he does have some really good stuff about that out there already. So I'm not stepping into your territory, Alan.
No, I actually don't think you are. I think everyone is helping complement each other, because sometimes different people need to look at the same information from different angles and different perspectives in order for it to click. I'm one of those people. I actually needed to use Alan's protocol in addition to yours to really there's so many self-doubting moments when you're going through the protocol of like, am I doing this the right way? It could just be how it's described. You really kind of go, I don't know exactly what that means. So let me read two protocols and see, are they both saying to do the exact same thing? What do they intend to accomplish here? So that's why I think there's value in having multiple variations on the same thing, and like hearing in your own voice your unique way of describing things is helpful. Like I said, I found your and this is not a knock against Alan, because like I said, his protocol is great, and it's also what helped you get to your version of it, and it's helping me to even look at it. And probably if I were to do mine, it would even be a more dumbed down version of it. It's even more approachable. Like the scientists are going to definitely have their way of doing it, and it's going to be very technical. Like my background's in software engineering, I could write something extremely technical for software engineers. But someone who's not a software engineer is going to write something very similar, but in more approachable general terms. So I think there's value in all of those things. So that's exciting, though. There is the possibility of discovering new species. Have you had a chance to do that at all? Like, have you? I guess one of the questions I have is, if you get a 93%, how do you know that it's not discovered? Like, who do you reach out to say to get confirmation of that? Like, let's say you want to publish a paper and characterize a new species just for fun.
Yeah, no, that would be super exciting. I mean, the first thing you have to do is you have to find it several times, because if it's just a one-time find, that's just not enough to sort of say this is a new species, because it could just be like a freak of nature or weird aberration. So you have to find at least, like 3 or 4 or 5 specimens of and they have to consistently deliver a similar sort of DNA result. I have discovered a new species, but I've done it not through DNA, but through microscopy, because like I cultivate dung fungi. It's one of my hobbies. I have a large amount of dung growing over there. It's very, and there's certain species that when you found something new, because like there's comprehensive books about what exists out there in the world, and if you find something that doesn't look like that, it's new. But I haven't decided to like write about it. I feel like I need some of the skills and some of the language and some of the background. However, it is one of my ambitions in life to do that eventually, and it'll be the same process with the whole DNA thing. You just talk to a lot of people who know their shit, and then you try and figure it out, and you try and figure out how to do it. And after many painful attempts, hopefully you'll figure out how to actually accomplish it. So
it's a goal of mine as well. So I'll support you. You can support me, and perhaps we'll co-author something together. Who knows?
Yeah.
Although I don't know if your new species will be available or if it's going to be found in LA, but I certainly could go look for it.
Well, it's a dung fungus, so you just have to look at a lot of dung.
Hey, well, that sounds like a shitty job, but no, I'm just kidding. Okay, cool. Well, that's awesome. So you've published your stuff from GenBank. You reference it from iNaturalist. That's amazing. I want to switch gears a little bit. You mentioned before that you've done some presentations on psychedelics before. What's your experience with psychedelics? What is your general philosophy on the usage of psychedelics, if you have any? Maybe you just talk a little bit about them a little bit.
Okay. Yeah, so that's a slightly awkward topic in that I stumbled into psychedelic lecturing by accident, because like I have a bunch of lectures that I give, on russulas And so this lady that I do workshops for, she sort of said, like, what else do you have? And I said, I have great presentation of dung fungi. And she said, dung fungi? My members don't want to hear about dung fungi. But don't some, psilocybe be growing dung? Can you talk about psychoactives? Can you do me a lecture? And I said, sure. And I just read a shit ton of stuff about it and, like really immersed myself in the matter and stuff. So at this point, I know quite a bit, but it's, I'm not an active, I'm not an evangelist for psychedelics. I mean, I think they're amazing from a cultural perspective, from a therapeutic perspective, and from a biological perspective. I think they're absolutely fascinating, but I don't know. I'm not an expert on it in the way that other people are, like Alan Rockefeller or Paul Stamets or whatever. So I think there's a ton of potential, obviously, in treating depression and treating addiction. And I think we're going to see a lot of work happening there, and it's already happening. There are companies like MindBloom that are starting to commercialize it. And obviously, the downside of commercialization, people will take something that's cheap or free, and it'll suddenly you're paying $15,000 or your insurance company, because it's guided therapy and safe environment, etc. So it can easily turn into just another blockbuster sort of treatment. But I think they work magnificently well for a lot of people. Yes.
This might be a personal question and you're not inclined to answer it, but have you yourself had a psychedelic experience before?
Yeah, I do really poorly on a lot of drugs because I get massive headaches and paranoias, and sadly this is one of them. I do really well with alcohol, however. Which is my favorite drug.
It seems like a very German thing of you to say.
Yes, no, I wish I myself had a better experience, but it doesn't do as much for me as it does for a lot of other people, unfortunately.
Well, no, you're not the first person that I have encountered that have had a similar experience with it as well. And I think that there's definitely a growing movement of acceptance in evangelizing the potential for treatment of what I would say is a new medicine, but it is not for everyone. And it's certainly an intense experience. And I think that if you're going to use it for treatment or medicine, this is again, my opinion, I think that your best chance of success in having a positive experience is going to be under a somewhat controlled setting with someone who is experienced, who can guide you through that experience.
Yeah, I agree. Yes. And a lot of cultures that have used these for hundreds of years do exactly that. It's not just a leisure sort of experience. It's very much guided and supervised. I agree.
Yeah, and I do think that is kind of one of the dangers to both the movement of broad acceptance. It's kind of the same thing happened with medical marijuana as well, is that there's sort of like they use the medical side of it as a sort of shortcut path to legalization, but it deters from actually the science I think psychedelics really do have the potential to change your idea of consciousness. And that in and of itself could be a gateway to having experiences that can certainly put you on a pathway to seeing the world differently. And that makes all of the difference if you're someone that suffers tremendously from depression and PTSD and addiction as well. You often need to have a very powerful experience to, I don't want to say overwrite, but counter the experiences that you've had that led you to those sort of regulatory, emotional, mental health issues that you're having. Right. And that is not something, given the power of it, it's not something that I take it kind of seriously, in the sense I think it should be broadly available, and I definitely advocate it as a solution. But I also think that there's a tremendous amount of power with the experience that should be respected. And so I just say that because there's, a lot of young people, especially, who are discovering it because it's so easy to discover it online, get your hands on spores and grow it. And there's even, I think, some therapeutic process in the cultivation of mushrooms as well, because there's something about the farming of it and tending to your crop and watching something blossom and grow. But I also just want to make sure that if you decide to embark on that path, that you treat it with the respect that it truly deserves. And as you mentioned, humans have had a relationship with it for thousands of years, and they've had shaman and the tribe doctor to kind of lead these ceremonies.
Yeah.
So it sounds like your interests are a lot more closely related to the study of the species itself, the organism, not necessarily.
They're fascinating, yeah. They have like weird biological quirks that are not found in other fungi, and figuring out like why do they cause these effects. That's still not a settled matter. There's all kinds of theories sort of blowing around why psychoactive fungi do what they do. So I mean, that in itself is like a wonderful sort of puzzle to be figured out.
It is awesome. What are some of the things that make it stand out a little bit different as an organism from other mushrooms?
Well, like horizontal gene transfer, for example, like psilocybin, that is in many different fungi. It's not just in psilocybe, it's like in nocibes and in pluteus and in gymnopolis and stuff. So like, why does this sort of gene cluster reoccur in all these only very distantly related species? Like, how the hell did that happen? And it's horizontal gene transfer, and it's what happens when two mushrooms kind of grow in the same pile of poop and they're sort of hanging out with each other. They're not having sex, they're just sort of hanging out, but like they're so close to each other that this sort of little bundle of genes actually manages to sort of cross from one hyphae to another, and suddenly that mushroom also has those properties. Which is sort of kind of mind-blowing to me, but it's such an evolutionary advantage that somehow once you have that property, it gives you a great advantage over other fungi. So that's why it's so widespread. It's like,
that's pretty cool. I wasn't aware of that. That's really neat. That's really neat. And there's people that study the phylogeny of these gene transfers to try to figure out, like, was there a common ancestor that maybe passed these genes on or something?
A lot of times it's also convergent evolution. Like nocibee also has psilocybin, but they invented it separately. They had the same good idea, totally independently of the psilocybes, and they just sort of came up with the same things. Like when two engineers or inventors sort of come up with the same idea, you go like, oh, okay, just great minds think alike. So that happens in fungi as well.
Well, and I know the same thing has happened with bioluminescent mushrooms. You've had multiple separate species develop the same underlying mechanisms to produce bioluminescence, which I also think is incredible because they produce the same wavelength of light. They use the same underlying enzymatic process to produce the light. It's pretty amazing when you kind of realize that.
Yeah.
Something in nature is putting together the pieces to produce these amazing genes.
Yeah, and as a non-biologist, I marvel all the more because I lack any kind of knowledge of biological processes. So I'm sort of discovering this stuff in my 50s. I'm like completely blown away.
That is awesome, by the way. So I applaud you for, like, you are very humble. I can tell in the way that you talk about things, but it's hard for us to have perspective on our own knowledge and our own experiences. But I would say you're probably more advanced than you realize in your studies, even though, I'm shaking your head no, but I think so. I mean, you've written a protocol for DNA barcoding. I mean, that's pretty cool. And yes, you could say you borrowed a lot of it from Alan, and that's true, but all of science is iterating on previous science. So I think that's pretty amazing. I want to get back to the dung fungi a little bit.
Sure, my favorite topic.
Yeah, well, I want to know about that because I don't know a lot about dung. It sounds silly to say that. I don't know shit about shit, but first of all, you live in Brooklyn. Yeah. The only dung I can think of is going to be human or dogs. So what dung is available in your area, and like, tell me about your fascination with fungus that grows specifically on dung and what you're finding.
Okay, so that's, yes, not dogs and not humans, very bad, because those are really smelly. The only dung I deal with is herbivore dung, which tends to smell like almost pleasant, I want to say. And I find that in Staten Island there's a park, and there's a park in the Bronx that has deer. So I just, pick up behind the deer. That's my source of dung. And I got into it about a year and a half ago. I did a summer class with Professor Don Pfister, who's a mycologist at Harvard University. And it was like a 1-week class. And we were supposedly studying fungi sort of in our environment. And it was really dry and there was no fungi. So he said, like, why don't you pick up some dung and see what's growing? And I went like, oh my God, I'm not one of those people. That sounds horrible. And I did it, and I got so hooked because they're just amazingly, amazingly beautiful. The actual organisms, the fruit bodies, as well as the spores. It just blew my mind. They're by far the most beautiful fungi that there are, as far as I'm concerned. So I started my little dung farm. I'm going to just show you some of my, I have a lot of this around the house.
Oh, wow.
They're just like these little Chinese takeout containers with a kitchen towel that's somewhat moist, and you just put the pebbles or whatever you want to call them on it, and then you wait a few days, and then stuff starts growing. And it's really small. You need like a needle, like a surgeon. You take your needle and you transfer it onto the slide for the microscope, and you look at it.
Wow.
And yeah.
Wait,
and
so that's the media. Wait, my mind's getting blown here for a second. So you go out to where there are wild dung samples, collect them, bring them back, and you put them in these containers. And you give it a moisture source, which is the wet paper towel. And you just wait to see what spores have dropped on the fungus and let it grow.
Well, they didn't drop on it. Like the animal eats, vegetation that the dung fungus has cleverly cannonballed some of its spores onto, and then it passes through the digestive tract, which is a tough environment for a spore because it's hot in there and there's a lot of gastric acids. But dung fungi spores are super tough and they survive. And then they come out on the other end, and then they're right in the middle of the most delicious sort of substrate that there is, and they have a head start ahead of the flies, ahead of the nematodes. The dung fungus can start growing right there and then, and it's a succession. You get one species immediately, and the other one takes a week, and then 2 weeks. I've had dung fungi that popped up like 90 days after I first started incubating. So it's a never-ending delight. It's like a foray in a box. It's the perfect hobby for when it's dry and there's no fungi in the forest.
That's amazing. That's a whole nother, I've never heard of that before. And it's really cool though. It's really cool. So you don't have to worry about sterilizing it or anything like that, because you actually want to maintain whatever natural spores are on it or in it.
Right.
And you just put it in an environment where that fungus can begin to grow. So what do you do after you start to see, do you wait for like cottony mycelia to show up on it and then you transfer it away, or how do you study it after that?
You don't really see the mycelium. It's usually just fruit bodies. Some of them look like flasks, some of them like cups, some of them are like little pill throwers with little berets on them.
Do they already have it? Do they already have it?
Some of them already have it, but sometimes oftentimes you have to wait. Some of them look like fur balls, and you just sort of, then you take them and you look at them first under my dissecting scope, which is this guy, and then at the compound scope, and then you take pictures of it with your phone, and then you post it on iNaturalist. And the great thing about it is you're always the first person to post the species, because nobody looks at dung fungi, so you're always like the mayor of that town because you found it first.
That's a great niche that you've carved out for yourself.
That's right.
You may find some dung that doesn't have a fruit body growing on it, but you may bring it back and it will fruit naturally.
Yes. Yeah. Almost always.
Really, almost always.
Yeah. There's like every dung has fungi on it. It's like a really good substrate for fungi.
Amazing. Well, yeah. I know that other mushrooms love dung, and you could, there's a whole market for buying dung just for cultivating mushrooms at home. But I had no idea that you could just go out and collect the right kind of dung. Could you though do like dog dung? If you could get over the smell, would that also would all dung, if you find it outside, would it produce fungus eventually?
That's a really good question. And I think in theory, maybe one or two, but the problem is like commercial dog food is very sterile. And you know, there's like, it has to go in before it can come out. And same with humans. I mean, I'd venture there's probably one or two things that would pop up anyway, but that stuff smells. If you have a family like me, you can't afford to offend your cohabitants, because I'd get booted out in no time.
I have a dog, so I can attest to that.
Okay.
But no, that's fascinating. So how long does it take to fruit? And do you have to give it air exchange, or can you just keep it in the container with the
2 or 3 days? You can just leave it in the container. There's enough air in there. It doesn't like run out of oxygen or anything.
And you'll just see a fruit body rise out of it.
Yeah, I mean, you put it under the compound scope, because oftentimes they're so small, like a half of, like a 1/10 of a millimeter or whatever. I mean, they're very small.
Oh,
yeah, some of them are bigger, but they're very, very beautiful. I have an Instagram feed, dung_fungi.com. If you're interested, that's where I post myself.
Yeah, definitely. Well, now that I have some more context and background to it, I think, by the way, you should do a whole YouTube series on this, because I think I've never heard of it before. I'm not going to say I've heard of everything in the space, because I definitely haven't, but there's not a lot of people talking about this. And also
nobody. I'm the only person talking about it.
You might be. And actually, I think it's a great way to get people outdoors introduced to it, because what I like about it is it gives you a target. It gives you another thing to search for, because it's one thing to go out. I live in Southern California. Mm-hmm. We talked a little bit about that. You don't, in my urban environment, it's kind of difficult to find fruit bodies, save for maybe the couple of weeks out of the year where it rains. You'll see them in the yard. But, having another target in mind, which is going out and looking for dung, bringing it back in a little container, I don't have to necessarily wait for the conditions to be perfect. I can kind of create them and push it along a little bit and study, create the specimen that I want to study.
During the lockdown, it was a lifesaver. We were all cooped up in our apartments going crazy. If you just spend an hour just zooming over your dung fungi, it's the most soothing thing. It's like meditation and therapy in one. It's very relaxing. It was a total lifesaver.
That is awesome. I'm 100% going to try that. I'm going to go hunting for dung this weekend probably. So that's pretty amazing. Anything else about mushrooms that makes you excited, that you genuinely love about it? Or anything that you want to share about, just advice to maybe people who are new to mushroom growing, mushroom hunting?
Right. I mean, normally I would say like join a club, because the best way to learn about them and to sort of figure out what to do with them is in the company of other people. And they're usually very interesting people. My mushroom club was founded by John Cage, the avant-garde composer. And it attracts, people from all walks of life. And it's one of the few places in my life where I truly intersect with very many different people that I don't necessarily would meet, in my job or whatever. It's just a really pleasant way to spend time. Unfortunately, that's not an option right now. A lot of clubs have gone to Zoom lectures and, a Zoom ID section. So, but like when circumstances allow, I would definitely say like join a club, because it's so much more fun. Anything else about mushrooms and how to interact with it? Like I said, iNaturalist is a wonderful way to learn more and have other people help you. And also it allows you to then go back and see what you've collected. And then the last thing I want to say about mushrooms is just they're so freaking beautiful. I mean, there's just something so amazing about them, something so ineffable. And they're so ephemeral, they're here one day and they're gone the next day. And there's something very special when you find a beautiful mushroom, because it's like in a few hours it'll be gone, and there's just there's something weird and magical about them. So
well, I agree with that. I want to plug your DNA barcoding protocol as well. How can folks find that? Maybe I can link to it on the EverymanBio website if you're okay with that.
Yeah, like sure. I mean, I always made it for other people to use, but I'm in marketing, but I'm not going to market my protocol. If people find it, that's amazing. But there's other, I'm not saying it's the best protocol. It's just my protocol. So yeah, but by all means, if people are interested in it, they should reach out to you, reach out to me, or whatever. If it works for them, great. If it doesn't work for them, I'm sorry.
No, I mean, I literally have used it, so I can say that it works. It definitely does work. I think that's also a sign of it being successful, that you're not marketing it at all. It's a Google document that you have to know the URL to. And the fact that people are sharing it amongst themselves, you don't have it up on any of your profiles. It's truly word of mouth. So that is a great testament to it actually being a great protocol. So one thing I like to do on these podcasts is ask if you could show us your lab. So by the way, you have a YouTube channel. Maybe you could share the name of your YouTube channel, because you've done I think it's 4 videos walking through the DNA barcoding experience using this lab.
Yeah, that was the pandemic, you know, when we were all sitting inside not knowing what to do, and there was no work. I was like, oh, I should do a YouTube series on my protocol. So yeah, that'll never happen again. I don't actually know the name of my YouTube channel, to my shame. You're going to have to remind me or post it or something. I don't know what it's called. It's called DNA Fungal Barcoding or something. It's 4 little videos that are literally just like, once again, meant to hold people's hand as they go through the process. And my lab, it's let's see if you can get a good look at it. So it takes up about 2 square feet, yeah, like this. And the sort of heart and soul of it is my thermal cycler. I'm going to sort of put that right there in front of me. It's this little baby. It's tiny. It's the cutest little thing, and it just cools and heats my template, my little tubes, and it's.
And that's a mini PCR. That's
mini PCR. Yes. There's definitely cheaper stuff out there. There's older stuff out there. Yeah, it's worked really great for me, but I'm sure there's many other things that work well for people. So I'm not going to plug it beyond that. I really like it. Then it's good to have a centrifuge, which is this guy. You don't actually need it. Some people do it without a centrifuge, but what it does do, it spins down all the sort of debris, the cell walls and all the things you don't want in your DNA sample to the bottom of the tube. So it's a useful piece of equipment. And then the last thing we talked about, it's my gel electrophoresis setup. It's really just this tiny little box. And if you look at a lot of labs, their setup is much bigger but much clunkier. And this is such an elegant little piece of design. It does everything you need it to do, and it's just very self-contained and lovely. I'm a big fan of it. It's very compact. I live in New York City, you know, I don't have a lot of space, so everything needs to be compact. And the rest is just, you know, tubes and pipettes and all kinds of other stuff. It fits into 2 shoe boxes. I don't have it out all the time. So if people think like a lab, it involves like setting aside a room or whatever, it really is not the case at all. You can just set it up on a coffee table and make it disappear again. It's very family friendly.
Yeah, your setup's great. You could almost put that in a suitcase and travel with it if you wanted to.
A rucksack, I've done that. Yeah, it fits into a rucksack.
That's very cool. And the reagents, where does someone go to get the primers, the tack, and the different solutions and reagents that they need?
Right. I mean, here's a plug for the Odin, which is an online shop sort of geared towards biohackers. It's kind of cool. But the reason I use that over other supplies is like most other supplies won't deliver to residential addresses, and they give you a hard time. They say, you know, what's your business address, and can we speak to your finance department? And you're like, yeah. So I've run into trouble there. So the Odin has no qualms about shipping to residential. In fact, that's their whole business model. So I really like them. The prices are not expensive. I do get my primers from a company called IDT, I think, where you just tell them what primer you want and they'll make it for you. And sometimes I use like slightly different primers, and they deliver to residential. So bless them.
And the lab that you use for sequencing, they're DIY friendly as well.
Yeah, I mean, so there's like the big guys are GeneWiz, and those are the ones that everybody seems to use, but they are really not happy about working with people like me. I had to spend a lot of time with their customer service saying this is all just for the New York Mycological Society and it's a business and it's all kosher, and they reluctantly so let me use them. But the other lab that I use most of the time, MC Lab, they don't care. They're really cheap. There's no customer support that I know about. You literally just send them stuff and they send you the sequences. And their order form is a bit of a nightmare to fill in, but they're very,
it's a spreadsheet.
Really yeah, it's a very complicated spreadsheet. I've done hundreds of sequences at this point, and they've mostly always deliver them, unless I fuck up, which happens occasionally. And they're really cheap. So.
And the reagents, you can keep those in your regular freezer, correct?
Yeah, I put them in a little plastic bag in a rack and put them in my regular freezer next to the steaks and, you know, whatever. That doesn't, the reagents don't seem to mind.
And it's okay to like take them out, thaw them, do the work, and put them back. No problem.
They don't seem to mind either. I used to worry about that, but yeah, I've had no problems with that, I think.
Yeah, and I think one of the great things about your protocol too is it says in there, it has your email address on it. It says if you like have questions about one, the protocol, and two, even the ordering process, because I'm about to fill out that crazy spreadsheet and I've got questions for you, are just like best how to organize your collection, like how do you label them and things like that. I think having your experience, having done it, will probably save me some time, because I'm a little OCD about staying organized and labeling everything nicely and having good schemas for tracking things.
Oh, same here. Yeah, I'm a neat freak.
So what is the best way for folks to get a hold of you? You mentioned your Instagram. Maybe you could plug that one more time. What's the best way? I know you've told me. I don't
underline fungi at Instagram.
I will link to that in the description of the video, as well as a link to the protocol, so we could try to get as many people interested into DNA barcoding, because you have one of the most, non-scientific, easy to approach methodologies that I think anyone who's interested in doing some DNA work, and come on, I mean, who doesn't want to extract DNA and sequence it and publish a sequence to GenBank? I mean, I get so excited by that. That's not a lot of people have done it, all things considered. So it is very approachable, very doable if you are interested in doing it.
Right. Yeah, and you can also totally publish my email. I'm more than happy to hear from people. In fact, I'd be thrilled to help, because I was that person once, and I did have mentors who helped me out. Damon Ty, Craig Trester, Anthony, like people that I could, call on when things like went sideways. And so I would definitely want to be that person to other people as well.
That is awesome. Sigrid, I'm so glad that we were able to find each other and connect. And thank you again for doing this and for the protocol as well. And I look forward to keeping in touch and seeing where your journey goes. You're doing some amazing things, and I'm just so thankful for you taking the time to meet with me and share this conversation.
Oh, no, I'm thrilled that you're interested. And good luck with what you're doing. And yes, we'll keep in touch, okay?
Okay, sounds good. All right, bye-bye.
Bye.